Good afternoon. We're going to get started. I'm Leon Platanias. I'm the director of the Lurie Cancer Center, and I want to welcome you all here. Before we start, I wanted to mention that we are very excited that we have President Seal with us in this session and showing his support for nanotechnology. Thank you for being here. And now we're going to move on with the session, and it is my real pleasure to introduce our session speakers, Dr. Priya Kumthekar and Dr. Alexander Stegh. Dr. Kumthekar is a certified neuro-oncologist and an associate professor of neurology and medicine at the Feinberg School of Medicine. Priya received her undergraduate degree from Kent State University and her MD from Northeastern Ohio University. She completed her residency in neurology at Northwestern and a fellowship in neuro-oncology also here at Northwestern. Dr. Kumthekar is a national leader in clinical trials. She is the principal investigator of 6 multi-center national treatment trials in neuro-oncology, and she serves as the neuro-oncology disease team leader and co-chair of the scientific review committee for the Lurie Cancer Center at Northwestern. She also serves as the National Executive Officer of Neuro-Oncology at the Alliance for Clinical Trials in Oncology. And in that role, she oversees the conception and development of clinical trials from early phases through the whole registration phase and development of the studies. Alex Stegh is a professor of neurosurgery at Western University these days. He used to be here. And he is the research director of the Brain Tumor Center at the Siteman Cancer Center, as well as an adjunct professor of neurology at Northwestern. Alex was born in Hermlein, Germany, and received his undergraduate degree in biochemistry from the Leibniz University in Hanover, and his PhD at the German Cancer Center in Heidelberg, and the Ben May Institute for Cancer Research at UFC. He completed his postdoctoral training at Harvard Medical School's Dana-Farber Cancer Institute, where he became interested in functional genomics. After he was promoted to instructor at Dana-Farber in 2007, he joined Northwestern as an assistant professor in 2009. With appointments at the David Department of Neurology and the Robert H. Lurie Comprehensive Cancer Center. In January, he was named the research director of the Brain Tumor Center at Siteman Cancer Center at Burns-Jewish Hospital at the Western University in St. Louis. So I would like to welcome them both here. Thank you.
Thank you, Leon, for that kind introduction. So we're going to ping pong a little back and forth between Alex and myself today. So I'm going to, for those of you in the audience who are not familiar with glioblastoma, I'll start by telling you a little bit about the disease, some of the challenges, and as you all know, with challenges come opportunities for us to forward the field in glioblastoma. Alex will then talk about how we're going to be thinking outside the box with nanotechnology within the field of glioblastoma. And then we'll come back to me and I will discuss our really exciting first-in-human phase 0 window of opportunity study that we published earlier this year. And then we'll tie it up by talking about where we're going to take it from here, that the next generation SNAs and what we have planned for the future. So for those of you guys who are unfamiliar, glioblastoma is a grade 4 primary malignant brain tumor. So this is a tumor that originates in the brain itself and not metastasized from the body. So really where it is in the brain is what determines what symptoms that a patient presents with. So if it's in the motor center or the speech center, we get, respectively those deficits, and then those are considered what we call localized symptoms. And then we also have generalized symptoms with glioblastoma. So those are things like headache, maybe personality changes, basically symptoms that occur because of increased intracranial pressure within the brain. So how do we currently take care of glioblastoma patients? After surgery or biopsy where the diagnosis is made, essentially it's a combination therapy of things like radiation, chemotherapy, and a little bit more of a newer therapy called tumor treating fields. Despite these aggressive multidisciplinary measures of essentially 4 different modalities, unfortunately, currently, the prognosis still remains quite grim at around 16-month overall survival from time of diagnosis. But not for lack of effort, right? We as a field within neuro-oncology are really working hard to try to forward the science here and really change these numbers that currently exist. To give you guys an idea, glioblastoma affects 12,000 patients in the US every year, which honestly might seem like a small number, but we were just talking last night in dinner, this affects all of us. I think many of us here have a story of a cousin, a friend, a brother. This affects so many people. And with 12,000 a year, it really ends up to being about, we did the math yesterday, around 400 a day. So this is really impactful to society. And with that, intervening, if you look at interventional and recruiting, actively recruiting studies on clinicaltrial.gov, you're seeing about over 300 clinical trials that are actively recruiting in this space at any given moment on average. If you compare that to something like invasive breast cancer, which is even more prevalent, we see comparable, if not maybe even slightly less, on the order of actively recruiting studies. So as a field, we're really trying to change these numbers and really trying to move that mark. So with all that effort, what are our challenges, right? It's important that we see, even with all this effort, what's slowing us down. So let's take a moment to go over these few challenges that exist in glioblastoma therapy. The first is the blood-brain barrier, and I think we've spoken about this earlier today, and I'm sure everyone here has heard about this in some way, shape, or form. The blood-brain barrier is important. As a neurologist, I can tell you it protects the most important organ in the human body, the brain, right? When we accidentally or purposefully ingest toxins, the blood-brain barrier is there to protect the brain from that. Unfortunately, it also protects the brain from therapeutics that we're trying to get in. And so this is something that when we are talking about treatment in glioblastoma, we need to talk about how to overcome this physical barrier. And many of you guys may have, as we alluded to earlier, there's a lot of people, even with that 12,000 a year, that we know affected. We've seen people in the news and the media affected, and we have a few examples here. These folks are diverse in so many ways, in age and gender and background, but yet, even through their differences in heterogeneity, they all succumb to the same disease. And what we've learned through these folks and many, many others is this is a really heterogeneous tumor. Genetically, it could be very, very different from patient to patient, and our therapies need to make sure that we encompass that heterogeneity. The third challenge that I want to highlight today is essentially understanding what's druggable and hopefully expanding upon what we can target and what is druggable. Traditionally, when we have learned about small molecule inhibitors, targeted therapies, these are typically therapies that target an enzyme or a receptor. But really, there's proteins that we're learning about within glioblastoma and beyond, and actually beyond brain tumors as a whole. We have proteins that we know are present in certain disease states, and we need to know and we need to develop methods to actually target those proteins as well. So 3 really big challenges that you'll see will come full circle with that we're able to highlight with future therapies. So with that, I'll turn it over to Alex.
Thanks a lot, Priya. So we believe that nanotechnology can really provide very unique tools to really address these challenges. And here at Northwestern, as a team of biologists, of clinicians, of chemists, material scientists, we decided to leverage nanotechnology to really reimagine drug discovery and drug development in the neuro-oncology space. And over the decade or so, we have really focused on one platform that we refer to as spherical nucleic acids, or SNAs. And I think SNAs really represent a prime example of a homegrown technology that really went through all the different phases of drug development. So from its initial discovery by Dr. Chad Merkin and Robert Letzinger, to preclinical testing, to clinical evaluation, and now to the generation of next generation as architecture. So, and that is really the story that we want to tell you today, where all of these different processes occurred on one campus under one roof. And that's really what this presentation is about. So what are SNAs? So when we talk about SNAs, we really talk about a design concept where we have a core that is surrounded by a shell of oligonucleotides. So these are small fragments of nucleic acids. And the important take-home message here is that SNAs represent a new form of matter, because nucleic acids in their spherical form, they have properties that are fundamentally different from their linear counterparts. And these include high stability of the oligonucleotide payload and the ability of these structures to penetrate cells and tissue very effectively. And in addition, our early data suggested that we can use these SNAs to dial down the expression of cancer-causing genes. So we refer to these SNAs as gene regulatory SNAs. So these are SNAs that carry a specific type of oligonucleotide. We call this an siRNA nucleotide. So based on these really promising early studies that we had, we decided to really drive SNAs, gene regulatory SNAs, towards clinical opportunity, towards patients. And what were really the critical questions that we needed to address here? Number one, what cancer-causing gene is a rational target for gene regulatory SNAs? Do SNAs reach the brain tumor? So what Priya talked about, the blood-brain barrier being a very robust biological barrier, are SNAs able to overcome this biological barrier? And once they are in the tumor, can they dial down the expression of our target gene of interest? And lastly, also very important for really moving these structures forward into clinical testing, are SNAs safe? So with respect to the target, we have chosen a target that we call BCL2L12. And we have chosen BCL2L12 for really two important reasons. Number one, BCL2L12 is overexpressed in the vast majority of GBM patients. So more than 90% of patients actually showed high-level overexpression of this gene. So this here on the left-hand side, this is an immunohistochemistry of a tumor sample, and everything in brown here indicates the presence of BCL2L12. In addition, we discovered that BCL2L12 is a fantastic progression marker, so we see the highest level of BCL2L12 in the most malignant form of gliomas, in these grade 4 tumors. And the other reason why we have chosen BCL2L12 is because it has a very unique multi-pronged mechanism by which it can promote GBM progression. So number one, we discovered that BCL2L12 is able to promote the growth and proliferation of tumor cells. And secondly, BCL2L12 is able to inhibit the activity of two pro-apoptotic genes, which are called caspase-3 and p53. And by doing that, BCL2L12 renders tumor cells resistant towards apoptosis or programmed cell death. So it prevents tumor cells from dying, either apoptosis that occurs spontaneously or apoptosis that is induced by radiation and chemotherapy treatment. Do SNAs cross the blood-brain barrier, which is really the most fundamental question that we very early on tried to address, and we were absolutely stunned by the results that we saw. So what we did in this experiment here, we took a glioma-bearing mouse, and we took these SNAs, these gene regulatory SNAs, and we directly injected them into these mice through an intravenous administration. And what we discovered was that SNAs can actually travel through the bloodstream and can actually accumulate in tumor elements. So that is shown here on the left. So this is an histological section of a tumor-containing mouse brain, and everything that you see here in this dark brown stain indicates the presence of SNAs. So yes, SNAs are able to cross the blood-brain barrier and can accumulate in tumor elements. And we also looked and analyzed the intertumoral distribution of SNAs. And that is really shown in here in this little video. So what you see here is a three-dimensionally reconstructed MRI scan. So you see here, this is a mouse brain. This mouse brain contained a tumor in the left frontal hemisphere. And everything that you see here in red, everything that you saw here in red really indicated the presence of SNAs. So SNAs don't really stay focused or localized to specific areas of the tumor, but they actually have the propensity to pervasively infiltrate tumor, which is really important if we think about gene regulation. Do these SNA have biological effect? Are they able to antagonize GBM progression? And we tested this in mouse models. So in order to generate a mouse model, we can take mice and we take human glioma tumor cells. So these are patient-derived tumor cells that we can isolate from a patient tumor. And we directly inject these tumor cells into the brain of these animals. And these tumor cells, we can call them glowing tumor cells, they are engineered to express an enzyme that produces a bioluminescent signal. So in essence, the more bioluminescence we see, the larger the tumor is. And you can see here in this middle panel that when we treat these tumor-bearing mice with L12 targeting SNAs, tumor volume is significantly reduced. And we can look at this over time, and you can clearly see here that our BCL12 targeting SNAs are able to really halt or at least slow down tumor progression. And this activity correlated very nicely with a very significant knockdown of our target gene, specifically in tumor cells. So what I've shown you so far is that we have a good target, I think a rational target that was preclinically vetted on multiple levels. We found that SNAs are brain penetrant, so we can use them as a systemic therapeutic modality. They are able to accumulate in the tumor. They can dial down the expression of our cancer-causing gene. And the last really aspect that we needed to address before we can move this into clinical trial is the question of toxicity. And to make a really long story relatively short, we performed toxicity studies in rodent and non-human primates, and these studies really revealed no significant toxicity associated with the treatment. So based on these results altogether, which took us several years to accumulate, we were now ready to really move these structures forward into a first-in-human clinical trial.
So yeah, with this preclinical background, we then moved towards the clinical trial. And I'll, in a further side, kind of get into some of the details of how we, from a regulatory perspective, got there as well. But the design was relatively simple. We had, in total, 8 patients that we ended up consenting on study. This was a non-randomized study where each patient was pre-surgically treated with NU0129, the compound that we've been discussing, and then went on about a day later to surgery. The primary endpoint of this study was safety, but also we were looking at drug accumulation within the tumor, and we had multiple secondary endpoints looking at pharmacokinetic studies, looking at other clinical endpoints, as well as biologic and target effect in the form of apoptotic markers. So this slide I could probably spend an entire talk on, but I think it's really important to pause and understand the steps that it took to move from the years of preclinical work that Alex and others did, and then to move it into the clinic with myself and my colleagues. So in order to do this, an investigational new drug application is needed, or an IND with the FDA. In order to do that, we need to make sure that the manufacturing process is clinical grade. So this has to go a GMP, or good manufacturing process. Of course, once we get the appropriate regulatory approvals from the FDA, we need that institutionally as well, with an institutional review board, scientific review committee, and then of course we need patient recruitment, and we can't forget paying for the clinical trial. I think Sam mentioned earlier that investment of really getting things from the preclinical work to the clinic seems like a small step, but one that really requires multi-level of investment. I always joke that this process, from when I started working with Alex and others to when we actually got the first patient on, in that period of time, I got married and had 2 babies. I mean, these things take time to, they take time to go through these hurdles and steps. And sometimes there's a back and forth where you go to the FDA, you have a package, and they might turn around and say, this all looks great, but this one thing's missing. So there's this back and forth, and understanding the nuances of this is extremely important. So once we finally did get our study going, we found that it was very safe. I'll also keep that primary endpoint relatively boring because we had no dose-limiting toxicities. We had nothing that showed that this drug was harmful to our patients. So moving on to understanding the drug accumulation within the tumor, we were very fortunate to have a great scientific collaboration with the Argonne National Laboratories. So here, what we did was we were able to, once we resected the tumor tissue, use X-ray fluorescent microscopy to then demonstrate whether or not these gold particles were uptaken within the brain and more specifically within the tumor cells. So what you're looking at here, what you're looking at here is actually a tumor cell. So this was as verified by our pathologist. And these are truly the gold particles that have been taken up. So we achieved overcoming that first obstacle of getting through the blood-brain barrier and actually getting drug where it needs to go, which is so much of the battle again in glioblastoma and other neurologic diseases. This slide is another one that I'm going to take a few minutes on because it's a little bit complicated, but illustrates a really important point to us. So here, what we have here are our patients who were treated with NU0129. So this is their tissue that we took at recurrence. So they were treated, and we took their tissue, and we were looking at the apoptotic marker caspase. What we found was that in comparison to these same patients, so actually I'll say this a little different. This is a timeline here where we have their recurrent glioblastoma tissue, and we compared that to, in black, their primary or initial glioblastoma tissue. And what we saw in looking at that timeline, comparing their primary, their first tumor resection tissue, compared to their recurrent tumor tissue, that we saw a rise in that apoptotic marker. We saw that appropriate biological response that we were looking for, which was, again, very reassuring. But we wanted to take this a step further and understand, is this just something that happens normally when a glioblastoma patient recurs? So as a control, what we compared it to are patients who had not been treated with this drug. So patients again at 2 time points, so primary, so their initial tumor resection, compared to their recurrent tumor resection. So these are patients, again, not treated with this drug, and comparing their initial tumor surgery, or their initial tumor to their subsequent tumor, we found that that same increase was not seen. So this served as our control, and again, was really encouraging to us that not only were we getting drug to the right place, but we were getting the intended biologic effect that we were looking for. Hand it back over.
So I think these data really provide important proof of concept that, number one, SNAs are safe and that they are a potential therapeutic modality for GBM. But like everything in life, GBM are incredibly complex. So research over the past decade or decades really illustrated that these tumors are on a genetic level incredibly complex. So in addition to the intertumoral heterogeneity that Priya was talking about, so we're looking at different patients, different tumors, and all of these tumors have somehow a different genetic makeup. In addition to this intertumoral heterogeneity, we also have to consider a high degree of intratumoral heterogeneity, which means that even tumor cells within the same tumor have different genetic elements that drive the progression of individual tumor cells. In addition to that, we learned of the concept of pathway compensation. So what does this mean? It means that when we try to target a biological pathway with a specific drug, for example, in this case, a drug against pathway component A, we try to eliminate this target, tumor cells find a way to bypass this blockage by activating an alternative pathway that creates the same biological output. So when we think about GBM therapy, or quite frankly for the matter, cancer therapy in general, we have to think about multimodal strategies that not only target individual signaling components, individual pathway, but that are actually able to target multiple cancer-causing pathways at the same time. And research over the past decade or so also has shown that immunotherapies have to be part of this conversation. And you may have heard of checkpoint inhibitors. So these are monoclonal antibodies that can activate the immune system to actually fight the tumor. And this discovery was awarded the Nobel Prize in 2018. And it was absolutely paradigm shifting because these inhibitors had phenomenal activity in certain forms of cancer, for example, in advanced melanoma. So when we treat patients with these checkpoint inhibitors alone or in combination, when we combine two different checkpoint inhibitors, we see very significant increase in long-term survival. And our former President Jimmy Carter, I think, is a fantastic example that really speaks to the success of immunotherapies. Now, the problem, however, in the context of GBM is that immunotherapies, including checkpoint inhibitors, don't work, or they have very, very little effect. And that is because GBM are what we call immunologically cold tumors. And that is in stark contrast to hot tumors such as advanced melanomas. So in melanomas, there are two things going on. Number one, they are characterized by a significant infiltration of T cells. So these are fighting immune cells that can attack and eliminate the tumor. In addition, these hot tumors also have a bunch of other tumor fighters, so other immune cells that can antagonize tumor progression. Now, in GBM tumors, which is a cold tumor, the situation is very different. Number one, there is a sparsity of T cells, so there's very little T cell infiltration in these tumors. In addition to that, they're characterized by a very high abundance of immunosuppressive cells. So these are immune cells that can actually promote tumor growth. So here's our thought about the next generation SNAs to really tackle this problem, to develop a multimodal treatment platform. So what should these SNAs be able to accomplish? Number one, they should accomplish to actually kill tumor cells. So very similar to what we've shown you with respect to our gene regulatory SNAs targeting a specific cancer-causing gene, they should drive tumor cells into apoptosis. But in addition to that, they should have some function on the immune system, some effect on the immune system. So they should be able to recruit and activate T cell fighters, and they should be able to convert immunosuppressive into immune activating cells. So how do we envision this on a molecular level? So this should be accomplished by inhibiting the STAT3 signaling pathway in all three types of cells. And it should be accomplished by concomitantly, at the same time, activating the SIGA-STING immune sensing pathway. So to those of you in the audience who are signaling aficionados, this is how it works on molecular levels. So STAT3 is a transcription factor, actually a master transcription factor. It is able to bind to DNA, to specific elements in DNA, which we call promoter elements. And upon binding to these promoter elements, it can drive the expression of genes that are associated with the promotion of tumor growth, the inhibition of apoptosis, and in immune cells by creating an immunosuppressive phenotype. So the idea that we had is, can we leverage SNAs to deliver oligonucleotides against small fragments of nucleic acids that are able to actually sequester STAT3 and prevent it from reaching target gene promoters and consequently stop STAT3-dependent gene transcription. At the same time, this SNA should also be able to activate the SIGA-STING immune sensing pathway. So SIGAS is what we call a DNA sensor. It is an enzyme that basically acts like a little policeman in your immune cells and that tells them, wait, there is foreign non-self DNA here, so we need to activate the immune system. And it does this by activating the downstream signaling component STING, and STING in turn can drive gene expression that are mainly associated with immune activation. So we developed these SNAs over the past few years, and we tested them in our mouse models. So in this experiment here, we use what we call a lukewarm GBM. So this is a mouse model that not perfectly recapitulates what Priya sees in the clinic. So these are not truly ice-cold tumors. So we call them T-cell inflamed tumors. So there are some T cells already in these tumors. And in this kind of lower-hanging fruit, we can see that our monofunctional STING agonistic only SNAs are completely sufficient to induce long-term survival in these animals. But then we wanted to challenge ourselves, and we really looked at a tumor model that we would consider ice cold. So again, that is really characterized by the absence of T cells and a large abundance of really these immune suppressive cells. And what we found was that, well, our monofunctional SNAs, well, they have some effect, but it's really nothing to brag about. But with our bifunctional ones, we can produce around 30% of long-term survivors. So right now we are working hard on these structures, really optimizing them, trying to increase these anti-tumor effects that we initially saw, and then most importantly combine them with checkpoint inhibitors to really see whether we can further enhance immune responses. Now, these experiments were done where we took SNAs and we directly injected them into the tumor. So this is a direct intratumoral administration. And we can argue this is a very feasible way to also administer these SNAs to patients, but granted, it is a very invasive way of delivering therapeutics. So again, we thought about a different way. Again, leveraging the increased stability of the oligonucleotide payload, leveraging the increased bioavailability of SNAs. So we thought about intranasal delivery. And the advantages of an intranasal delivery are clear. Number one, it's very easy. These drugs are typically very easy to administer. And it allows us to really dose the patient multiple times. And most importantly, it is non-invasive. And it has been shown that drugs that are given intranasally can actually reach the brain. So there are right now approximately 80 clinical trials out there that investigate drug delivery using intranasal delivery. And what many folks have found, that intranasally delivered drugs can actually reach the tumor or can reach the brain through a nerve pathway. And that is actually precisely what we see with our SNAs in our tumor models. So when we isolate nerves and we look at the distribution of SNAs, we can actually see that SNAs can really travel along these nerves and that way get access to the brain and the brain tumor. So again, this is illustrating to you that nanotechnology especially, it allows us to really consider, for example, in this case, routes of administration that are typically not achievable with conventional drug design. So with these structures, we really get privileged access to sites that are typically non-accessible. We tested them, so we tested the nose-to-brain delivery of the Sting monofunctional SNAs, and we can clearly see an effect. So again, we use this glowing tumor cell model system. With our Sting SNA, we can dampen tumor progression. So in essence, with these intranasally delivered SNAs, we get good tumor control. And that is another path forward for us to really work out the specifics. How do we have to design SNAs, and how do we have to optimize this specific way of administering drug to see efficacy?
All right, so I'm going to wrap things up here and maybe go a little bit faster than what I was planning because, you know, I know we discussed a lot of things today and I'm very excited to see what nanomedicine and nanotherapy does for the future, not just of glioblastoma, of many other neurologic diseases. But I'm going to skip this slide and really just pause here and tell you what I think what I really want to demonstrate to you conceptually. The prior slide was more a science wrap-up. But I think strategic and conceptually, this slide is really what I want to speak couple sentences to. So what you've seen in our last 20 or so minutes is just this really amazing collaboration between the clinical side of the world, neuro-oncology, neurosurgery, blended with chemistry, biology, immunology, and I very much credit Chad for breaking down these barriers and many others in this room. And it's sort of my plea and my strategic concept to tell you guys, this is really how I think we're going to make a difference in glioblastoma and other diseases, is to continue to break down these walls between our various schools and campuses and locations. And I'm so glad to come up from Chicago today from our medical campus to talk to you guys about this, and come down anytime. We can grab a cup of coffee and talk more about how we can break down these walls. And I think that's really what we wanted, Alex and I really wanted to demonstrate to you all today, and tell you that we're looking forward to more kind of getting rid of these silos and having all of us come together in a synergistic way. So thank you so much.
Okay, we have time for a couple of questions. Anyone who wants to ask a question, please, let's get a microphone there. Yeah.
This is absolutely fascinating that SNAs can do eventually break that PBB barrier, which nature has put such a mechanism. My question is that once it reaches SNAs to the brains, what eventually happens to this one? Once it has done its job, eventually does it lead the system, or it is there? Is there any danger for that if it remains in the brain?
So as far as our maybe I use this. Maybe okay. As far as toxicity studies are concerned, so we've done toxicity studies that lasted approximately 4 or 5 weeks, and in that time frame and at the dose administered in both rodents and non-human primates, we did not see any adverse side effects. So maybe Priya, you can comment on some of the sort of side effects that we saw that relates to skin discoloration, etc. So they were relatively minor. In rodents, we looked very carefully at clearance as well, and really at biodistribution as a function of time. So what we discovered was that typically we see SNAs accumulating in the brain as early as 4 to 5 hours after we give them intravenously. And then slowly they decay with respect to brain accumulation. We looked, I think, at mice 4 to 5 weeks after SNA treatment, and we see some residual SNAs that are left, approximately 20 to 30% that have not been excreted from the body. We also looked in patients. So when Priya showed you the timeline, so we looked at the first recurrence. So typically, or in most cases, patients recur again. So there's a second reoccurrence. So we isolated those tumors as well, and we compared them to the first recurrence, and we looked at gold levels in the second recurrence. And to our shock almost was that these tumors, they recurred, but they still had gold in them. So even after several months in a patient, there was still gold present. But I think that is something we can use for our advantage, because we still have basically a therapeutic left in the brain. There's no oligonucleotide, but there's gold. And gold, for example, is a fantastic radiosensitizer. So we can think about when we re-irradiate these patients, that there's still gold present, and hopefully that will have, an additional therapeutic benefit. And that's something we would be really interested to.
Actually, Chad.
I really want to ask a question. First of all, incredible presentation. It was absolutely wonderful. Our president said very accessible, which is a good litmus test. But the question I had is, you laid out this whole incredible, I think, game plan to take something, from crude from the benchtop into the clinic. And it's exciting that we got there. But it's also frustrating that you said, 2 kids and a lot of time in between. What is the biggest holdup? I mean, you talked about the science, regulatory, is it money? What would accelerate? Because you've got such a disease that has, such a horrible impact on the world. Yeah. And even though we're working very, very hard, we're still, in some respects, working at a snail's pace.
Yeah, no, I think that's a great question. And I'm excited even for future collaboration, just for a do-over with some of this, because I think we've learned a lot through these processes and can work on the speed. But I think, to answer your question shortly, we need all of it. So we certainly need the money and the investment and the collaboration, whether it be from that investment or from working with industry companies, to make sure that early on we're engaging the regulatory bodies as early as possible, and then doing, all preclinical studies are of interest, but really strategically guided preclinical studies that hit the point that the FDA wants to see. And hitting those early, having those pre-IND or pre-IDE meetings early, whether it's a device or a drug, to then make sure that we are hitting those exact preclinical studies in a strategic fashion, and that speeds the process along. So certainly financial investment, early engagement of regulatory, and then very strategically posed preclinical studies to get there fast. Because I think we all love the science, but it needs to be directed, and then we can do the ancillary preclinical studies as we need, but just focusing on the ones that are needed for quick for quick regulatory acceptance.
And if I may add, Chad, there is a conversation of cancer centers across the country right now to talk to FDA oncology to decrease some of the process there. This will happen. There is a meeting being scheduled, actually. So, any other questions? Go ahead. Yeah.
Thank you. Thanks for the great talk. My question kind of related to the previous one. Because we've seen how much great work, tremendous amount of work that went into the research and going through the regulatory process. I'm wondering, as scientists, do you have any say on, once it came out of the market, the availability of this medicine to people, for example, family, friends of graduate students who probably worked on this? It's probably going to be incredibly expensive. Do we give our hope to the private sector and the government for subsidizing it, or we have any control over, I don't know, the availability, I guess?
That's a big question and an awesome one. I'm going to attempt to take it, but I want to tell you what my, at least, range of expertise is, because your question is really about commercialization and accessibility in terms of it's almost a health insurance question. And so it's a little bit probably beyond the scope of my individual knowledge and in terms of expertise. But what I can say is we're working, I think having partnerships with industry is really important, right? Because I think that leads to fast commercialization, which is, I think, partially embedded in your question. But then we aren't the ones that set the price. And so that's a little bit more complex. So maybe we circle back to that later. But I think ultimately that commercialization process, we need to work with our industry and pharma partners to make it as smooth and as accessible as possible.
Great. Okay, was there one more question there, or yeah? Okay.
I'm not sure the feasibility of this, but have you guys looked into switching out the core of your SNAs instead of being gold to being a more an easier material for the body to break down, like silicon or silicon carbide? I'm not sure on the binding process, though, there with the RNA.
Yeah, that is something that we're actively working on right now, because when we think about translation, I think a gold core is not the right way moving forward. I think we used it deliberately simply because it really allowed us to track the distribution of these structures very precisely. And that was really important in the first proof of concept in a patient. So we needed to really understand where they are at any given time, do they reach the target, etc. But moving forward, you're absolutely right. So we need more biocompatible materials. We have shown both on the gene regulatory side, so Chad has been done enormous work on other cores. They work very similarly on some of the immunotherapeutic SNAs that alluded to. We have proof of concept that the core doesn't matter, it's the three-dimensional architecture that matters. So we can switch out the core and, for example, use a lipid nanoparticle, liposomal, or PLGA-based core. So that is really right now the next step in terms of really getting these into the next clinical trial.
Okay, one more question and then we'll have to move.
So thanks for the presentation. What type of sequences you use to inhibit STAT3 and treat GBM? So
there are two types of sequences that we use. We use classical decoy oligonucleotides. So these are short double-stranded DNA that are derived from a STAT3 binding sequence in a promoter region in some of the target genes. In addition to that, we use G-quartet oligonucleotides. So these are forming structure that bind basically to the SH2 domain of STAT3 and prevent it from docking onto other signaling molecules. So kind of two different types of oligonucleotides.
Okay, great. Thank you again for a great presentation to our speakers.